Hello, and welcome to this Nature.com custom webcast titled U-Disco for Whole Organs and Organisms, a New Way of Large Sample Imaging. My name is Jayshan Karpan and I will be your moderator. Today's webcast is sponsored by Bitplane, Levision Biotech and Andor Technology and will be given by Dr. Ali Erturk of the Munich University Hospital. We'll begin today's webcast with a presentation and end with a Q&A session where we'll address the questions that have been submitted by you. To ask a question, just press the Ask a Question button at any point during the webcast and we will answer them today. Over to Ali.
Hello everyone, my name is Ali Erturk. I'm the head of Acute Brain Injury Research Group at the Institute for Stroke and Dementia, which is a part of Ludwig Maximilians University of Munich. Today, I would like to talk about a new technology that we have developed recently that is called U-Disco, which is a tissue clearing method. Before going into details of it, I would like to briefly talk about why tissue clearing is an important technology and what it can be useful for. Neurological diseases are a major health problem, especially in developed countries. For example, stroke and dementia alone represents 1/3 of all deaths in high-income countries. Annually, about 7 million people die from neurodegenerative diseases. Even though there isn't any effective treatments, the available care and medications cost about 250 billion euros annually alone in the EU. This number actually excludes the burden of family members, both suffers financially and psychologically. In addition to neurodegenerative diseases, about 10 to 30% of the developed societies suffer from neuropsychiatric disorders such as schizophrenia, addiction, or depression. Overall understanding how the brain is wired will lead to the way to unlocking the most powerful tool, the human brain, in health and diseases. However, as you would also appreciate, human brain is the most complicated being in the known universe. Even a mouse brain, which is routinely used in experimental research, consists of 75 million neurons, and maybe with billions or trillions of synaptic connections. While the nervous system is extremely complex, neurons form highly organized networks which can last even throughout one's lifetime. So, mapping the brain overall makes sense and potentially a doable task which will lead to understanding of diseases like neurodegenerative or neuropsychiatric issues where the brain wiring is affected, either it degenerates or the wiring changes. To give an oversimplified analogy, you can think neuronal networks in the brain like the networks of transportation systems with all roads. Mapping the brain is similar to mapping the roads, which can help to understand the traffic and repair when something goes wrong. In the real world, there is about 30 million kilometers of roads in total. If we can interpolate the expand of a neuron, let's say 100 kilometers of road in real world, so mapping all roads in the world would be like mapping only 0.2% of the mouse brain. So to map entire mouse brain, we need to map a world 50 times larger than the current one. That is a lot of work, but it's doable, especially with the tissue clearing methods that we will talk shortly. Well, actually, a human brain is about 2,000 times larger than a mouse brain, and it's estimated to contain 100 billion neurons. Therefore, the overall task is much larger, of course, but still, I personally believe that it is doable within a few decades. To map the human brain, already there have been quite a few initiations, especially in the US and in EU, spending about $1 billion to map the brain, or at least let's say to start mapping the brain. And also Japan, Canada launched their own human brain mapping projects, and China and South Korea are also planning to start their own programs. So it is clear to everyone that understanding how brain is wired and how it works is really critical to, of course, to see how we can think, how we can learn, as well as how we can treat the diseases like Alzheimer's, Parkinson's, or depression or schizophrenia. So the major tools so far have been to map the brain. On the one hand, MRI, fMRI, or DTI-like methods, which you can see the gross anatomy, the entire brain, but not very high resolution. So you don't see individual neurons, you just see the gross morphology. You see the cortex, parts of cortex or hippocampus, but you don't see individual neurons. Good thing about these approaches, you can of course get information on live human being and follow them over time. On the other hand, there is serial EM methods where one produces thin slices of the tissues and then map in very high resolution. So you reach the synapse level, even the more detailed than synapses, you can see individual vesicles and details of cytoskeletons with this method. Very high resolution, but the issue is that it is very laborious and time consuming, and just you can achieve imaging about a millimeter size of tissue already in several months or years, and to map only mouse brain may take 50 to 100 years using this method. So today I would like to talk about maybe a better solution, that is tissue clearing, where you can visualize the gross anatomy of the brain, so entire brain you can visualize, and you can visualize it at single neuron level. You can look into individual neurons, their axons and dendrites and their connections. And it is very time and cost effective compared to other methods. So potentially this method is the one closest to map the human brain or the mouse brain, probably as first, anatomically. Now I would like to briefly talk about our major research interest in the lab. We are interested in understanding traumatic brain injury. Traumatic brain injury has a high incidence, about 240 per 100,000 people, similar to AD cases, both in the EU and in the US. And it actually mostly affects developed countries because of traffic accidents and sports accidents. Also, the total cost annually is very high, about $100 billion both in the US and in the EU. And similar to other neurodegenerative diseases, there is currently no effective treatment for TBI. So as you would know, of course, it starts with an acute injury to the brain. An external force affects the brain, and then there is acute degeneration at the lesion site. The tissue there disappears. And when you follow actually people in the clinic, what we see is that this degeneration in some cases spreads to distal parts. Some people, they have thinning of cortex away from the lesion. That tells that there is a global change in the neural networks in the brain. And there is also associated problem. Those are secondary degeneration or chronic degeneration, and this causes early onset of dementia, epilepsy, psychiatric disorders, and some other non-neurological problems. So people with TBI, they suffer chronically over years from different diseases because of the spread of this initial injury from the lesion site to distal parts of the brain. So to model TBI in the brain, we use controlled cortical impactor device. And with this device, you can define the parameters and apply the same injury on different animals. And using the same parameters, we found that actually there is two different types of injury. Either injury is mild, little, as you see in the MRI here, or it's relatively large, moderate, or severe in some animals. And we actually, now I need to mention, we do this injury close-up, skull is intact. And we found that actually the animals having large lesion is usually, actually in all cases, the ones having the skull broken. When the skull is intact, it's protecting the brain, and resulting lesion is very small and mild, as you see here. Also, several weeks later, the tissue is quite preserved in mild case and degenerated in the moderate case. And we also confirmed this with IgG staining showing the blood-brain barrier leakage, very little in the mild lesion and very extended in the moderate lesion case. So using this injury model, then we wanted to study the pathophysiology of the TBI in the mouse and see what happens to neurons and also glia cells. First, what we did was to follow the animals with moderate TBI using T2 weighted MRI. You can see here that at the 4 hours injury time point, the lesion area has this edema, this white signal, and you can see over time it's kind of dissolving, and it's starting to be like a cavity degenerating. You can see the black hole here, and in 4 weeks the cavity is larger, expanded, and combined with ventricle. Therefore, it looks white because it's now filled with CSF. And it seems to be stabilizing over weeks, and quantification shows the same. Here is first these 3D images, the edema in yellow, red is the degenerating tissue. You can see edema is dissolving over a few weeks while the degenerating tissue is forming and being stabilized around 1 to 2 weeks, and it's not expanding. Unlike the human case, we were expecting that this tissue will start to degenerate, the distal parts of the brain will also degenerate, but we didn't see it at the MRI. And we asked the question then, are we missing here something? As you know, MRI only shows the gross anatomy. Then we did more detailed imaging using confocal microscopy, where we performed this lesion on Taiwan GFP, and animals where a subset of neurons are labeled with GFP, therefore we can see fine structures of neurons like dendritic spines. We then quantified dendritic spines in TBI case. First, you can see here 1 week post-injury from the ipsilateral side, ipsilateral hippocampus. You can appreciate the clear reduction of dendritic spines compared to unlesioned case. While if you look at the distal parts of the brain, the spines are preserved. There isn't really significant change also shown in here in the quantification. And that is 1 week post-injury. And if we wait long enough here, 1.5 years post-injury, pretty long, you can see that the decrease in spine density in the ipsilateral hippocampus is preserved. But surprisingly now we are also seeing changes in distal parts. Away from the lesion, there is now decrease in spine density, also in contralateral hippocampus compared to unlesioned controls. Next, we check the neuroinflammation in the brain using IBA1 immunolabeling, which stains microglia and macrophages. And we could see that in 1 week post-injury, the microglia is accumulating mostly at the lesion site. This is original data, and this is the heat map view. And this is 1 week post-injury data, and this is unlesioned control. And you can clearly appreciate that microglia, they are accumulating around the lesion mostly. What was surprising for us, 1.5 years post-injury, so 1.5 years later, the neuroinflammation was not disappearing, but we could see a widespread of inflammation now evenly throughout the entire brain. And when we look at these individual microglia and macrophages in the brain from lesion side or so distal sides, they look also reactive microglia and macrophages. They had retracted processes and also enlarged cell bodies compared to unlesioned controls here. This is 3 months, this is 1.5 years post-injury. So it looks like there is a chronic neuroinflammation, which is also associated with chronic neurodegeneration, which is more evident at dendritic spine level. And based on this data and many other data I skipped today, we believe that acute brain injuries such as traumatic brain injury or stroke, they induces persistent structural and molecular changes in the brain, not only at the lesion site, but throughout the entire brain. So entire brain is being affected over months and years. Then our major research interest is to understand the mechanisms of this spread, how this local degeneration spreads over time to distal parts. What are the mechanisms of this chronic neurodegeneration after acute brain injuries? Of course, this information will be very important to develop strategies to block this spread. Then we can rescue the development of the chronic problems like early onset of dementia or neuropsychiatric problems. We could inhibit these chronic complications in TBI patients. The major problem in experimental research now then how to accurately assess changes in long neuronal circuitry throughout the entire nervous system, throughout the entire brain, how we can see this local degeneration is spreading to distal parts, changing the distal circuitry. If we were to use traditional methods like tissue sectioning, when we want to make hundreds and thousands of sections of tissue, image them one by one and stitch them, put them together to make a mapping of the brain to analyze the circuitry, we know that this is very, very laborious and very time consuming, and it's mostly not complete because of distortions during cutting or staining or imaging. Then also putting all this data is almost impossible on the computer. Ideally, one has to image the brain without sectioning, but we know that the light would not go through because the tissue scatters the light. So only we could image the surface of a tissue. But we and others developed new technology that is in general called tissue clearing methods, where the biological tissue rendered transparent. Now the light can travel end to end. We can visualize the inner structures of the organs, the individual cells throughout the entire thickness. Just to mention some of the prominent tissue clearing methods that have been published recently, and those are mostly water-based methods like scale, or there are also organic solvent-based tissue clearing methods, the disco methods that we developed, and also now there's the hydrogel embedding based methods like Clarity and Pact. And the list is actually now growing more and more. We also have to maybe mention the expansion microscopy related research, which uses some of the concepts in tissue clearing like hydrogels, and then using them to expand the tissue, then look into tissues in high resolution. The work initiated by Ed Boylan's lab, and now picking up also in different dimensions that we can visualize details of cellular structures using this actually sophisticated method. Now we are looking at a different perspective. Instead of expansion, today I will talk about shrinkage, which we call shrinkage microscopy, SHM. That is developed by us, ultimate disco or U-disco clearing method. In shrinkage microscopy, what's happening is that tissue shrinks into about 1/3 of its size. Actually, this may be not very, very dramatic, but actually it's very sufficient to help a lot in imaging large organs and organisms. For example, when you clear the whole mouse body, usually you cannot take it into microscope because the mouse body is really much larger than the chamber and sample holder of the microscope of the standard light jet. But when it becomes 1/3 of its volume, original volume, we could now fit it into the light jet microscope that we bought commercially, and we could image the entire mouse with light jet microscope from head to toe. Now I would like to step back a little bit and talk about the initial disco, which is called 3D-disco, 3D imaging of solvent cleared organs. Why we developed this method? So during my PhD work, I was working on axon regeneration in the spinal cord, and we were following the axons using live imaging. We could see the axons in sensory axons on the surface of spinal cord before, and then we would make lesion, and then they would, after some treatments, manipulations, they would start to grow, regenerate. But what would happen is that they would go into the spinal cord, they would dive in to the ventral part, and then they would disappear. We could not see anymore with live imaging. And if we do histology afterwards, take out the tissue sections, then now we were looking at only fragments of axons. That would not tell us really, is it regenerating axon, or is it a spared axon? Is it the ending or a part of middle part of the axon? So all these problems were really causing issues. Therefore, I guess many studies claiming to induce regeneration in the spinal cord could not be reproduced because of these methodological limitations. When you look at fragments of axon and claim that you caused regeneration, that is actually a big question mark, whether you are really seeing the regenerating axons or spared. So with this shortcoming in mind, we wanted to do something better to overcome this problem and really study regeneration in an unbiased way. We started clearing. We tried different protocols. We ended up developing tissue clearing method that is called 3D-disco based on THF and BAP or DBE. Using this method, we studied regeneration of axons after conditioning lesion in the spinal cord, and then we could define their trajectories throughout several millimeters. Now I would like to show you a short movie about some of the achievements with 3D-disco method. So this is type 1 GAP animal, the spinal cord, you can see individual axons and motor cell bodies. Now this is a scan through spinal cord, entire depth with 2-photon microscope, not only light sheet, but you can use other microscopes like 2-photon, and you can see really high resolution details through entire thickness. Then you do 3D reconstruction, and now we are able to follow individual axons over several millimeters. So here I'm just showing you a small part of the scan, but we can do this over really whole spinal cord. And now you see larger segment of spinal cord where there is the regenerating axons after conditioning versus unregenerating axons where they are stuck. And in the regenerating part, you can see that they dive under the lesion and go up again. So that technique really became very useful to study these trajectories. So if you want to learn more about this, please go ahead and check our detailed protocol or original paper published in Nature Medicine. We are happy that shortly after our publication, it has been used in many studies and people get results in different research fields such as immunology, stem cells, or cancer biology. Within just a couple of years, there have been quite a lot of publications using 3D score and getting results. So overall, 3D score was very useful to clear the spinal cord. It worked very well with the antibody labeling. It kind of worked for mouse brain, but didn't work very well. The signal wasn't so great. One issue was that the fluorescent after 3D score clearing would not survive very much, in the order of a half-life a day or less. Then we could not see the details like dendritic spines because they would go away easily. And 3D score actually would not also work for whole body clearing, as I will show you. To overcome these problems, especially to clear the mouse brain or entire CNS of a mouse, to look at big picture in traumatic brain injury case, as I mentioned, we were interested in studying how the local degeneration spreads to the rest of the brain or to the rest of the CNS. We started working on 3D score to improve it. And in the lab, Chen, Marika Francesca, and Aliza took this job, and they end up developing a much finer version that is called Ultimate Disco or U-Disco, where the fluorescent could survive several weeks to months. Therefore, we could clear actually not only individual organs, but the whole body of the mouse, where you could see the signal throughout the entire body. We will go through this in a short time. So first, let's see how 3D scope perform on whole body clearing. Well, we started with our good old method, 3D scope on Taiwan GFP animal. Here's before whole body clearing with 3D scope, and this after. Clearing looks not bad, nice, but when we look at under the microscope, there is no signal coming from the brain or spinal cord. Here are the zoom images. You can see that very, very dim signal, if any, survived. One reason is that the chemicals that are used in 3D scope were not really proper to keep the fluorescent proteins, actually the signal coming from endogenous fluorescent proteins over a long time. Here I have to make this clear. This signal coming from the dyes, like Alexa dyes or nuclear stainings, they survive fine, but the proteins, endogenous, the expressed proteins like GFP, EGFP also would not be surviving. One of the major reasons is that, for example, tetrahydrofuran that is used in dehydration step of 3D scope is prone to oxidation and further reactions. One of these chemicals that are used in the limitation part, refractive index matching part, that is BAP, benzyl alcohol, and benzyl benzoate, the other component of 3D scope, they can form peroxidase and quench the fluorescent signal as well. In contrast, in U-Disco now we are using terbutanol in dehydration step, which is more stable and less likely to do oxidation or the other kind of reactions. And in the refractive matching step, we are using DPE. In DPE, actually this is a solution that is containing DPE with BAP, So DPE has less reactive. DP actually doesn't have this reactive benzylic CH or CO bonds, as you can see here. So these are here in the benzyl alcohol. You can see this kind of or DBE. These bonds can react, and in this chemical in DP you don't have them. Therefore, DP is less prone to radical changes, radical reactions, or other oxidative reactions. Finally, we also use antioxidant vitamin E to scavenge further the peroxides that can destroy the fluorescent proteins. So how UDisco performs? This is the brain after UDisco whole body clearing versus 3-disco whole body clearing. You can appreciate the signal throughout the brain. In UDisco case, very nicely preserved all around, while in the 3-disco case, you can see only some signal left in hippocampus where there is a lot of expression. You can see the quantification of total intensity very low in 3-disco compared to UDisco. And also the contrast is very, very little in 3-disco compared to UDisco. You can see normalized signal to background ratio. You can really clearly see the signal in different parts of the brain. You can separate axons and dendrites from the background really easily. So we perform UDisco whole body clearing, and this allows us also to do this 10 millimeter scans from ventral part of the animal and dorsal part of the animal with light chain microscope. Then we combine them and get whole body scanned mouse. So here also I want to mention the usage importance of tissue shrinkage, because adult mouse has about 2.35 centimeters of chest width. And this is actually, if you have that much thick tissue, you can never image with the commercially available objectives, which has maximum of 10 millimeters scan. If you want to combine ventral dorsal, you can image only maximum 20 millimeter tissue. So after UDisco clearing, these tissues over 20 millimeters, they become smaller than 20. Now we can really scan without missing any tissue in the middle from dorsal, ventral, then combine them. We have whole body scanned mouse. And these are some parts from different organs, different body structures, the brain, here the other organs from the mouse and from the rat. And this is like a whole chest area where we have, in a single scan, we have the images from the lungs, from the liver, from the intestine. And this is view with the corner cut where we can visualize now here the cells that we transplanted. An important point that I would like to discuss a little bit is UDisco actually preserves the ratio of the tissue in different dimensions, ratio of the individual organs. So some organs actually can have a little bit different shrinkage. Clear example is the bones. They shrink far less than the rest of the tissue because they are heavily calcified and they are strong, hard. But the parts that we are interested in, many other people are interested in measure the brain, spinal cord, the CNS tissue actually is very even. You can look at your brain before clearing, after clearing, when we enlarge this shrunken tissue and put on the before image, you can see it very nicely overlaps different anatomical regions from the brain, and the ratio is actually 1-to-1 in this the ratio of the size of the different tissue layers. So it's not really changing. The gross anatomy is preserved. If we look into individual cells before and after clearing, that is also preserved. And here is the neuron before clearing, and here is after UDisco clearing. When we enlarge this and put on top of the before, it perfectly matches. And the same for microglia and vessels. Actually, there is really significant overlap. And this is the shift in the order of actually sub-micrometer or a few micrometers. So compared to 50, 100 micrometers of tissue. So in the order of less than 5%, if any, there is change. And here is now a view of whole body of a mouse after UDisco clearing. This GFPM mouse. You can see that the clearing is nice. You can see throughout all the tissues. And you can also here appreciate how the volume is really reduced. As I mentioned, the bones are not really shrinking. Therefore, the mouse has more or less similar length, but the volume is now about a half or a third. That helps us a lot to put it into this tiny chamber of the microscope and visualize it by moving the animal in this orientation. We image the head and the chest and inner organs, and it goes on. So let's look at this cleared animal under the microscope. This is just a perfluorescent, really just a perfluorescent image of the cleared GFPM mouse. You can see that the brain is lit. Even the area with hippocampus where there are more cells are really bright, and the spinal cord is also quite clearly visible. The neurons that are expressing GFP. So now we will look in more details using light sheet microscope. So now we take this animal intact after whole body clearing with UDisco. Now we will look through light sheet microscope. So first the head, and this is the head, really head, and the bones are, you can see the is intact here. Here is the brain, and you can see the nerves going to the eyes. Let's zoom in. The ones going to the eyes, the ones going to the whiskers. So basically you have really individual axon resolution. So subcellular resolution, you can see tiny parts of the axons. You can see even tiny fibers of axons everywhere in the head. Now look at the brain. Is after clearing the brain, and you can see nicely the neurons throughout the entire brain. You can see the axonal tracks. Now we will zoom in. Here's the cortex, the neuronal cell bodies, their extensions, and the axons going into the middle brain, midbrain, and here's the hippocampal area, the hippocampal neurons, and the corpus callosum. You can appreciate even the tiny fibers of the corpus callosum axons are almost you can resolve them with this method. If you were to zoom in more using a high magnification objective like 20x, actually you can see now individual dendritic spines very nicely throughout the entire tissue. Now we are traveling on the animal down. We are looking to the spinal cord on animal. You can see the individual dorsal root ganglia, their extensions in the spinal cord, and the periphery, and then also all the neuronal extensions in the spinal cord. Now look at this clearing of the leg. Just I wanted to show how that is clear very nicely. You can now appreciate the axons throughout the entire limb. Those are the axons going to the toes. You can see fibers and the ones going to the muscles. Not only the adult mouse, but you can actually image the adolescent rats. Here, a rat that is 200-300 grams that is cleared with UDisco, and transparency is very high and very clear. You can see. So there's a really large animal, about 10 times of a mouse, more or less close to the size of a human brain. And we labeled actually the vasculature in this animal with using lectin dye conjugated to FITC. And then we can see that the vessels throughout the overview, right, you can see the vessels, the organs with heavily vascularized. And we will zoom into the vasculature in the CNS, rat CNS, intact rat CNS, brain and spinal cord. This is tissue of more than 13 centimeters. You have the details all around from head to the spinal cord. You can see really large vessels, small vessels. If you're interested in studying angiogenesis, vessel deformation after injuries or in neurodegeneration, this is really a cool method, and you can apply it in any organism level. Rats, mouse, and would work with other animals too. So you might say, well, all sounds cool, looks nice, but what do we do with this? What can we use this clearing method for? So I would like to tell about that. So if you ask me what are the applications, I can tell you that it is unlimited. It's just limited by our imagination. I think if you just sit back and think 5 minutes, you will come up with 5 different ideas how you can use this in your research. I would like to give a few examples. For example, we use this method to screen the transplanted cells in host mouse in an unbiased way. And what we did was to check where the bone marrow stem cells are going in the host animal. Apparently, it is known that when you transplant these cells, they go into the lungs and livers, the primary flow organs, but where else, it's not very clear. So to do these experiments, we get bone marrow stem cells from beta-actin EGFP mouse. We also loaded them with quantum dots that we have also a second color to visualize them more carefully. Then we did either IV or IP injections. Then we sacrificed, did whole body clearing and screen the whole mouse and find individual cells wherever they are. And in control case, PBS injection, there is no stem cells observed as expected. In IP or IV case, there is these cells already, a lot of them in the liver and lung as expected. But what was surprising was that we see the individual bone marrow stem cells now back in the bone marrow of the host animal. Here you can see these individual ones. Not surprisingly, these cells then, they weren't in the CNS. We didn't observe them in the brain tissue. They couldn't cross the blood-brain barrier. An important feature of eDisco, like 3Disco, it also works very well with the antibody labeling, but in mouse or human tissue. Here on the left, we have the mouse tissue from Taiwan GFP-M animal, where we did immune staining for GFAP over a millimeter tissue, and here also cell body labeling with topo-3 dye. You can see very even nice labeling throughout the entire thickness. Details of the astrocytes are very clear everywhere. The same with human tissue, post-mortem human tissue coming from a patient who suffered from diabetics. You can see the astrocytes evenly stained throughout this thick tissue. You can observe the details of them. Now next I would like to show you a few videos to illustrate what we can really achieve, what we can really see in a way, but we cannot quantify it well. Hopefully that is next thing that we are interested in solving. This is the spinal cord and brain, so CNS of an adult Taiwan GFP animal. So now we will look through and travel into it. Let's start with the brain. Now we are coming close. You can see the neurons in the cortex, their extensions. Now we will go to hippocampus, cell bodies, and the axons in the corpus callosum, the axons coming from cortex. Now these are the cortical layers, layers 6, 5, 4, and so on, different layers. And you can see nicely cortical tree of the brain. Now we are back in spinal cord. You can travel in the spinal cord like millimeters, several centimeters till the end of the spinal cord. This is just a short segment, and you can easily trace any single axon that you're interested in. Please go ahead and watch the complete videos in the supplementary part of our paper published this month in October 2016 in Nature Methods. Next, I would like to show you another example. I would like to show you not only neurons, but now this is clearing from CX3-CR1 EGFP mouse, where the microglia are labeled with GFP. And important thing here is GFP expression in this animal is very, very low compared to type 1 animals. So there's another way to check the quality of UDisco protocol, how it works in also animals with low EGFP expression. Now we will travel into this brain that is cleared with UDisco to visualize individual microglia throughout the entire brain. Now we are coming close. You can see hippocampus, cortex, inner brain part. You can very nicely see every microglia in the whole brain. Now we have a perfect tool that we can go and quantify the numbers in different regions and analyze them in different cases in neurodegeneration, in Alzheimer's mouse, in stroke mouse, in TBI mouse, and so forth. So we have this data that we image the whole brain, whole spinal cord, whole mouse. But what do we learn from them? How do we analyze? What kind of biological questions we can answer? That actually should come now more and more. So we have to focus more and more how we can analyze this data. Of course, now we have to do not only single animal, but some that we can do statistics, image them, then get this data, find the ways to analyze them, and then maybe register them to some maps to get some data quantification of signal in different brain regions, compare treated case to untreated, disease case to healthy case, and so forth. Of course, all this needs also a lot of computational power. Nico Renier in Magtasier-Lavin group, who also published the original iDisco paper combining 3-disco method with immune staining to do whole brain immunolabeling in adult mouse. They recently published this new work, iDisco Plus, where they describe a method that is called ClearMap. What they did was to register the scans of the whole mouse brain into Allen Brain Atlas, and then they could get a readout, like microarray, like a readout, where you can see now the numbers of cells, and here in this case the plaques in different regions of the brain. So a work that is published together with them a week later in Cell Reports. They were staining the A-beta plaques and microglia and phospho-tau in the Alzheimer's mouse model at different time points. You could see that it is accumulating over time in different brain regions, and they get such a data over time, different brain regions, how much they contain these stainings. I think this is really a good start. Definitely we have to build on top of this. We have to get more and more quantitative approaches to get different tools can work in different conditions. But I think this is already a pretty good start. I would like to conclude my talk by giving you some summary about UDisco. So UDisco is a robust tissue clearing method. It is very straightforward and can be completed within a few days. And actually it does not really require complicated imaging systems or complicated clearing procedures. And it can deliver really high resolutions. You can look at the subcellular details of neurons. You can see individual axons, dendrites, dendritic spines in whole brain or in whole animal. And I believe overall UDisco will greatly help the histological studies. Then we can better study the organisms in health and diseases. And I also believe that an important perspective of this method, the shrinkage-mediated UDisco method, that it will help us to clear and image the human brain maybe earlier than any other approaches. At the end of the talk, I would like to thank my group, which actually made this work to come true. So particularly Chan, Marika, Liriza, and Francesca, they were the primary researchers creating this work, UDisco work. And well, I'm happy to take now your questions. Thank you very much.
Thank you for your presentation, Ali. It is now time for the Q&A. This is your opportunity to ask Ali your questions. To do so, all you need to do is press the Ask a Question button on your console, type in your question, and then press Send. So our first question, Ali, it asks, can UDisco be used to image cells, structures within bone?
Yes, actually we can use it. Also in the paper we have actually a couple of figures that's on bones, and we compare UDisco to other existing clearing methods from the bones that are used for bones. And in a way we are happy to report that UDisco achieves really good clearing over the bones, and you can see really through the bone and identify the cells in the bone marrow, as we also showed in the paper, the stem cell transplanted bone marrow stem cells after transplantation, we could identify them. Of course, these bones are from mouse, and if you use larger bones, it might get more difficult to clear, like the rat or even larger ones. So one has to try. Maybe then you need longer incubation times, but overall it's possible.
Excellent. Thanks, Ali. And our next question, this one's from Elizabeth, asks, does the shrinkage impact your ability to resolve fine structures?
In theory, yes, because there is shrinkage, and the shrinkage is about 30% in one dimension. That means you lose 30% resolution. Structures comes closer that amount. So what happens though is that after clearing, because we clear, we remove the background, we reduce it very significantly, we reduce the light scattering very significantly. In a way, we don't really feel this 30% loss of resolution after clearing. For example, we also demonstrated that we can see dendritic spines, which are actually already very small, about 1 to 2 micrometer in size, and after shrinkage they get even a little bit smaller. And with light sheet microscope, which has actually at most that much resolution, we could resolve them. That was also very surprising to us, because we could really see fine structures. But of course, that you are then most limited with the resolution of your light sheet microscope, that is mostly in axial that resolution. That is where actually major limitation comes, not really shrinkage, because shrinkage is not that huge in one dimension.
Excellent. Thanks, Ali. Our next question comes from Nicola, who firstly wishes to thank you for a very interesting talk. But she was also wondering that UDisco obviously shows a big advantage for endogenous fluorescence imaging. And she wants to know that the main purpose, but Does it also bring advantages for when you use Alexa dyes? And can this method benefit immunolabeling techniques as well?
Okay, so I will maybe go a little bit answer shortly.
Yes.
So yes, you can use the UDisco with antibody labeling. Alexa dyes, and if you want to do some secondary or also use DAPI or some other nuclear screening, you can use it. But overall, if it has some advantage over 3Disco, I cannot say it. I think there isn't really, because both really clears and allows you to see the signal, and then because both of them does not quench the signal of this kind of dye, so basically you can use any of this. And considering that 3Disco takes less time, maybe it might be even more advantageous to use 3Disco in some instances to observe the antibody labeled tissue.
Excellent. Thanks, Ali. And our next question asks, have you worked on brain tumours using this technique?
So we didn't, but one can easily use it, no problem. You can clear and image anything in the brain. Of course, it's important how you label the brain tumors. It should be fluorescently labeled, and this fluorescent expression should be relatively high. If you're using GFPM, cherry, whatever, you should make sure that you have good expression. For instance, just make simple controls, go to confocal without clearing, make histology. Can you see your signal coming from tumor easily with confocal microscope or epifluorescence? If you say yes, and most probably then it should work well with UDisco.
Excellent. Thanks, Ali. And our next question is from Lily. It asks, UDisco seems to preserve GFP very well, but she was wondering, how about conventional fluorescent dyes that stain nucleus?
Yeah, I think I answered this question. There's no problem, and also in the paper, figure 6, it shows that we have top row 3, DAPI works well, Hug staining or Alexa dyes works perfectly, no issue.
Excellent. Thanks, Ali. And the next question is from Eva. And she's a frequent user of clearing techniques and light sheet microscopy, and it's been working well for many purposes, but she's now facing a problem. She's trying to quantify volumes of the light sheet data, and in your talk you've shown heat maps of reactive microglia for brain sections. She's wondering if it's possible to generate these for sort of huge stacks of light sheet data.
Yeah, this is what I talk through the end of my talk, and this is just expanding field, and this is now another field of science, computational science, where we scientists should come together and use this data that we generate, others generate, and then make the algorithms that can go through this data and construct us these heat maps, or some easy ways to get meaningful information out of this data. And I mentioned the clear map from Nico Reiner from Mark-Tessier Levin Lab, which seems to be a first really significant step forward. And I am hoping that there will be other approaches which will present alternatives, and then this field should definitely expand.
Great, thanks, Ali. And our next question asks, what kind of computing power do you need to work with these large images that you're producing?
We are using, at the moment, actually workstations that we just get commercially, HP workstations with 256 gigabyte RAM now with, I don't know, multi-CPU with 12 cores, I guess. There are, of course, computer clusters in many universities, institutions. We don't use them, and I think even with them it can be much easier to analyze. But I think even the ones that we can buy commercially is okay for us at the moment to handle this kind of data, which is in the order of, let's say, 100 gigabyte for a mouse brain, and if we consider whole mouse, it's maybe a terabyte or so. And this is not only about the computer power, but also it's also software. Some softwares are easy to handle the data. They don't need a lot of computer power. Some softwares they need more computer power and RAM. And we are also communicating with software developers that they open the way that softwares, without pushing the hardware too much, we can analyze this data. So it should be both hardware and software hand to hand to analyze the data in future.
Great. And I think we've got time for just one last question, and this one's from James. And he asks, is it possible to mark specific targets with immunohistochemistry after clearing with UDisco?
Usually the immune staining is really done before and of clearing, because for immune staining you need a water environment and like PBS and so on. But disco clearing removes the water. After disco clearing, you don't have any more water around. If you drop your clear tissue into water solution, it will become opaque and probably will have some fractures inside because of the change. So, sorry, but actually, it's not really doable thing. So some people, I guess, tried, but I would personally not recommend doing immune staining after clearing, but just do it before.
Excellent. Thanks, Ali. Well, I'm afraid that is all we have time for today. Remember, you can watch this webcast at any time on demand at nature.com webcasts. I would like to thank Ali Erturk for his presentation and for answering your questions. The webcast sponsors Andor Technology, BitPlane and Levision Biotech. And I would also like to thank you, the audience, for taking the time to be with us today. Thanks for watching, and I hope you'll join us again for our next webcast.